Okay, good morning, everybody. So, welcome. As I'm sure you're all familiar with by now, agings and endings is where we deal with two main themes, with cancer and with the nervous system. And in the A cycle, the focus for the nervous system is predominantly on peripheral nervous system. You've had a couple of lectures about membrane potential and axons and how action potentials propagate. Today we're going to talk about how we actually initiate an action potential. Okay, so you understand about when there's an action potential, how it's going to keep moving. We're going to talk about how that gets started. We're going to look a little bit at what happens when there's damage to a nerve. You have a case for the Colles fracture. We're going to look at carpal tunnel syndrome, and you'll actually do a lab on that next week. And you've got some shoots to help you understand the action potential, because we know that this is a fairly complicated part of physiology, putting all of these things together. Okay, so if you've done AEB, you'll know about central nervous system transmission. I'll make a few comments about how this is different. If you haven't, then you need to pay a little bit more attention. So, the neuromuscular junction, the most studied synapse in the body. One downside of that is that there's lots of specific terminology, which probably if we were inventing it today we wouldn't come up with. I think we would just call it a synapse. There's nothing about the neuromuscular junction that's not a synapse. It just happens to be the name that was given to it. Okay, and it's logical, but it's kind of redundant to have a name for what's effectively just a synapse between a motor axon. Here you can see some axons coming down, and the muscle fibers, which are these red bars here. Kill those lights a little bit. Make much difference? No. Alright. So, key thing to know about the neuromuscular junction is that each muscle fiber gets only one synapse. Okay? When you're a baby, when your nervous system is developing, that's actually not true. You can have multiple synapses on one muscle fiber, but there's a process of competitive elimination. And so by the time you're an adult, less than 2% of muscle fibers have got an innervation from more than one axon. Okay, and you'll learn about the motor unit, which is what controls muscle contraction. That means the one axon coming down, that axon might actually innervate several hundred muscle fibers. So whenever that axon fires, we're going to activate a whole bunch of muscle fibers, but each individual fiber listens to only one particular motor axon. You can see obviously that there's some quite specialised structure here. When we pull the membranes apart and stain them separately, here on the left we've got staining of the axonal part, here on the right we've got staining of the muscle fibre surface. You can see that that specialisation exists in both cells, okay, in the muscle cell and in the nerve cell, and that they match. Okay, so this shape here on the axon is matched by this shape here on the muscle. These bits don't match because this is the actual axon coming down. The myelinated part is not making a synapse yet with the muscle. It's this part here, which is the neuromuscular junction. Okay, and it's very complicated and elaborate machinery. It develops over time, and it needs constant communication to stay healthy. Okay, so there's actually exchange of trophic factors. If your axon is severed, your muscle wastes away because the synapses are not maintained. They need constant communication down the axon and back from the muscle to the axon to keep this matched specialisation. Okay, if we look at what that actually looks like in a diagram. We can see there's a pretty elaborate structure there. We've got, on the axonal side, we'll usually call that the presynaptic side, so before the synapse. There is communication both ways, but the information flow is generally coming from central nervous system to peripheral nervous system down to the axon terminal, the presynaptic side, jumping the gap to come to the muscle, which is the postsynaptic side. Okay, so postsynaptic membrane, muscle, presynaptic nerve terminal. The whole point of synapses is to provide a space where there's chemical communication between cells in the nervous system as opposed to the normal electrical communication. Okay, so travelling down the axon is all electrical. You heard about from Dr. Hahn. When we come down here, there's a tiny gap, 50 nanometers, where the chemical transmission happens. After that, it's electrical again. Okay? In the central nervous system, that chemical component is essential. That's where we actually integrate information, where we can bring inputs from different neurons together. At the neuromuscular junction, I think it's fair to say that the chemical component is pretty much redundant. Okay? If we had just a direct electrical connection here, we would effectively still be fine. Okay? And that's the big difference between the peripheral nervous system and the neuromuscular junction and the central nervous system. Central nervous system, we don't want a synapse to cause activity on the postsynaptic side every time it's triggered. Okay? Every time you see an ice cream, you don't want to go and buy it and eat it, or you become morbidly obese in a couple of hours. What you want is the ability to modulate that by whether you're hungry or not, what else you've got to do, how much money you've got. But every time you decide you want to move your arm, you need your biceps to contract. Okay? There's not any other factors to weigh there. That's already happened up in motor cortex. So what we've got here is an unusual synapse where every time the presynaptic side is active, there's going to be a postsynaptic response. Okay? And that explains the specialized structure that we're going to look at. So, basic idea of a nerve terminal is that we've got vesicles. These are packaged full of neurotransmitter, these small blue dots. They can be docked, which means that they're ready to be released, or they can be fusing, undergoing exocytosis, releasing the neurotransmitter, which can then diffuse across the gap and bind with the receptors on the postsynaptic side. The region where the vesicles are docked, ready to be released, we call the active zone. Okay, so here's an active zone. A neuromuscular junction, there are many active zones and many docked vesicles, and we'll see why that is in a minute. Okay, so the basic process that happens at the neuromuscular junction is the same as at any synapse. Okay, so this is something that you definitively need to know and remember because it comes up in the autonomic nervous system, central nervous system, neuromuscular junction. So we have an axon that's been bringing an impulse from somewhere. It could be from spinal cord, it could be somewhere else. That's done by sodium channels opening. You guys are experts on that. When it gets to the terminal, the same process continues. The depolarization causes opening of voltage-gated sodium channels, causes some depolarization. Okay, the first different part happens here, that we've got voltage-gated calcium channels in the membrane as well as voltage-gated sodium channels. Okay, we need the sodium to make sure there's enough depolarization to open the voltage-gated calcium channels. Calcium comes in, works as a second messenger, and it's going to trigger the fusion of these vesicles that are sitting here with the membrane, releasing the neurotransmitter to the outside. Okay, a different neurotransmitter at different synapses, but calcium is always the second messenger. So at the neuromuscular junction, this is going to be acetylcholine. Central nervous system, it's often glutamate, but it could be dopamine, it could be GABA, an inhibitory transmitter. Wide variety of different transmitters could be released. They need to diffuse across that gap, the synaptic cleft, needs to bind with a channel, and this is not a voltage-gated channel, that's a ligand-gated channel now. Okay, it's gated by a chemical binding to it. Once these have their neurotransmitter bound to them, they open, lets depolarization into the postsynaptic cell. That depolarization will open voltage-gated sodium channels, and your action potential can run away down to the next step. Okay, so that sequence is always the same, that we've got voltage-gated sodium channels, voltage-gated calcium channels, we've got some processes that cause vesicle fusion, release of neurotransmitter, binding of the transmitter to a ligand-gated channel, it opens to let sodium in again, and we've got depolarization spread again by voltage-gated sodium channels. If we look at one of these synapses at the neuromuscular junction, this is a freeze fracture technique where you can slam an active synapse down onto a metal slab that's cooled down to minus 200 degrees Celsius or so. The idea is that you instantaneously stop biological processes. This is looking at the presynaptic membrane. So it's as though we're the muscle fibre looking up. It's not active, and what we see, these bumps, we think that they're calcium channels. Okay, because remember the membrane is a bilayer, but poking out of it are all those transmembrane proteins. And the prominent ones at the synapse, and particularly the neuromuscular junction, are sodium channels. When we stimulate the neuromuscular junction, so it's going to release, then we see this. Okay, and what's happening there is that we're actually seeing the vesicles. This is looking up into a vesicle. Remember, this is the presynaptic side. So we're looking up into that cavity. Okay, so here we are looking into a cavity as a vesicle gets released. There's quite a lot of machinery in these terminals. And so one thing that the system does, it doesn't actually let that vesicle go away. It fuses, then it actually pulls back and reforms as a vesicle, keeping its specialised apparatus. Okay, we'll look at that in a moment. So there's very clear evidence that this is how it works. What's useful from this slide compared with the previous one is that the calcium channels here are lined up with the active zone. Okay, so the textbook picture here is not actually very good. The calcium channels are shown up here. Really, the calcium channels should be focused down here where the vesicle release is happening. We don't need the calcium high up in the terminal, we need it right where that fusion process is occurring. Okay. So let's have a look at the vesicles, the presynaptic side, to get started with. So these terminals are quite a long way from the central nervous system. Okay, they belong to a motor axon. That means that that neuron has got its cell body inside the ventral horn of your spinal cord, and if it's reaching out to control your thumb, then it's spreading a metre or so. Okay, so the terminal is a metre away from the soma. It would be pretty tedious to have to make the neurotransmitter up in the soma and transport it down, so usually we actually synthesize our neurotransmitters in the nerve terminal. Okay. The peptides are too complicated for that. They actually do have to be transported down from the soma, but the non-peptide transmitters are done down at the nerve terminal. So here we see it at the neuromuscular junction. We've got choline to be combined with acetyl-CoA. There's an enzyme that does that, choline acetyltransferase. That's making acetylcholine inside the nerve terminal. We now need to get that acetylcholine inside a vesicle. Okay, and the way that we do that is we need transporters. We have to have an active process. So here there's a pump. Okay, this particular protein is using up ATP to pump protons inside the vesicle. Okay, so the inside of a vesicle is a bit acidic. We're pumping protons inside there, and then we use the proton gradient to drive a transporter. So as it lets a proton out, it exchanges that for an acetylcholine molecule being taken in. Okay, and of course there's a particular gradient that we can maintain with those guys. So we'll pack the vesicle until the gradient on that proton pump can't support it anymore. So the vesicles are actually reasonably standard in how full of acetylcholine they are. Once they're loaded, they need to be brought down to be docked at the active zone. That's not particularly the best picture, so they need to be brought down. There's a cytoskeletal scaffolding here. They travel down to be docked on the active zone, ready to fuse. Okay, so they can be sitting there but not fused yet. And then the other thing that this slide shows is that we need to preserve these pumps and exchanges. So once it's fused, the vesicle reforms, keeping these transport proteins inside it. Okay, so we've talked a bit about filling up the proton, using the proton pump, talked a little bit about getting it ready. Let's talk a bit about how the fusion side of things happens. So the vesicle not only has the transport proteins in it, which can load it up with acetylcholine, it also needs to contain some specialized proteins that are going to help it to dock and then to fuse. Okay, there's a wide variety of these proteins. They've got different names, which you don't need to remember. You need to remember the general principle, which is that they're called snares, okay, which is kind of handy. That does, by some strained thing, spell out to a very long chemical name, but everyone calls them snares. And if they're a V snare, they sit on the vesicle. And if they're a T snare, they sit on the terminal. Okay, and so here we've got synaptobrevin. That's an example of a V snare. Down here we've got syntaxin, which is an example of a T snare. And when the vesicle is docked, it's got these two proteins entangled with each other, holding that vesicle close to the membrane. Okay, because if you think about it, it's not much good us thinking, I want my biceps to contract. You send a command down, depolarization, calcium comes flooding in, and then you have to wait for a vesicle to crawl down the cytoskeletal scaffold. You need to wait while it gets close to the membrane. By the time that's happened, that calcium gradient's probably gone away. So what you need is to have this protein sitting here as close to the membrane as you can. You know, 10 nanometers away or less. Then, when the calcium comes in, and we saw in that previous electron micrograph that the calcium channels are sitting here very close by, we get a high calcium concentration, then we can trigger fusion of the vesicle with the membrane. Okay, again, the textbook picture is not very snappy, I don't think. So we know that this synaptotagmin molecule is involved in the fusion of the vesicle with the membrane, so they should really have drawn it down here. Okay, it's got a calcium sensor on it. The the calcium entry triggers this to pull the membrane that little bit closer. When the membranes touch, they fuse, you get a pore opening and the neurotransmitter comes out. Then you actually need to spend some energy to disassemble this, to let the snares unravel, so the vesicle snares and the terminal snares, and you get the vesicle floating back free, ready to be filled with transmitter. Okay, so this same process is happening in your autonomic nervous system, in the central nervous system, different transmitters, but that same basic principle of moving vesicles close, docking them, having a calcium sensing molecule to trigger the formation of the pore, and then recycling of that vesicle. There's lots of toxins that can attack this process. Tetanus toxin attacks the synaptobrevin, which is this one. Okay, that gives you lockjaw. Botulinum toxin attacks the snap, which is again involved in the fusing part of the vesicle formation. Botulinum toxin, or Botox, is used clinically. It's got quite a few legitimate uses. So if you have a child with a strabismus, a lazy eye, you can actually use Botox injections to paralyse the muscles on the side that it's being pulled towards, so the eye comes back to centre. Some people have got muscles that are constantly in tetanic contraction or spasm. You can use Botox there, and by attacking the SNAP25, you prevent vesicle fusion. If you can't release neurotransmitter, the muscle can't contract, and so the spasms go away. Because the terminal is so far away from the soma, it takes weeks or months before you get enough SNAP25 back in the terminal for that muscle to start contracting again. Okay? It's also used by Hollywood celebrities now to try and get rid of wrinkles, and the way it does that is that it stops small contractions of facial muscles, so your face relaxes and the wrinkles go away. Okay? The downside is you can't lift your eyebrows up anymore or do anything else because you paralyse those facial muscles that made the wrinkles go away. Alright, so that's the presynaptic side. Postsynaptic side, we've got a whole new cell. The plan is kind of the same, whether it's central or peripheral synapse. We've got to have a ligand-gated receptor, okay? And it's the neuromuscular junction, it's the acetylcholine receptor, the ACHR. Most of these ligand-gated channels have got 5 subunits, so this is looking down at it from above. There's 2 alphas, a beta, a gamma and a delta. And what they do is form a pore through the membrane. Okay, so what's shown here is one of the subunits. This is the alpha subunit of the acetylcholine receptor. It has some alpha helices that span the membrane. That's this M1, 2, 3, 4. It's got some extracellular component where it can actually bind the ligand, can bind the acetylcholine. And by bunching these up, like wagon trains drawn up to resist the attack on the plains, you form a corral where you can keep the hydrophobic lipids out, and you form a pore that can let aqueous type molecules through, so ions, in this case sodium, potassium, that sort of thing. If we look at it in cross-section on the right, you see here we actually have 2 binding sites for acetylcholine. We've got a pore running through the middle. The pore has got some negative charges in it, and that's what gives it its selectivity. Okay? That means that positive ions are attracted and can pass through, but negative ions that enter the channel are repelled by these negative charges. This is the selectivity filter, so they don't travel through. Because it's got 5 subunits, it's fairly big, so it's not very selective between sodium and potassium. Okay? the voltage-gated sodium channel's only got 4 subunits, it's got a smaller pore, it can start to discriminate between different cations. Okay, and if we look at what happens then when we record, this is recording on the postsynaptic side. We can either look at spontaneous activity, what's happening when the muscle's just sitting there. We see some little depolarizations. When we stimulate, we see some depolarizations, and they occur at particular sizes. Okay, this is using a very weak stimulus. We've somehow stopped that neuromuscular junction releasing hundreds of vesicles. We're getting it to release only a few at a time. Okay, and what was found when these studies were done is that you get bands. You get a band here at about, in this case, 0.4 or so. You get another band here at about 0.8, another band here at about 1.3 millivolts. Okay, and that was deduced to mean each vesicle that was released, the neurotransmitter from that opened enough channels to produce a small depolarization of, in this case, about 0.4 of a millivolt. Okay. That process we call quantal release because we never see a depolarization that's 0.1 or 0.2 of a millivolt. We never see a release that's 0.5 of a millivolt. It comes in particular quanta, and we think that the quantum is a vesicle. So you can release 1 vesicle or 2 vesicles or 3, but it's not possible to release 1.5 vesicles. Because we pack them roughly equally full of neurotransmitter, that's why each vesicle gives us roughly the same size depolarization. Okay, and the spontaneous release is, they're sitting there docked, there's hundreds of them, periodically the calcium's going to fluctuate, one of those vesicles fuses and releases its stuff. Okay, it's a waste, but that's just how the system works. To be sensitive enough to be fast, it needs to have occasional spontaneous release. So, this is the summary of what we've said. The vesicle is packed full of acetylcholine, somewhere between 5 and 10,000 molecules, concentration of 150 millimolar or something. When we release one of those vesicles, causes a depolarization of about half a millivolt. That's actually pretty similar to what happens in the autonomic system, central nervous system as well. What's special here at the neuromuscular junction is that a normal action potential causing a normal potential, okay, centrally this would be called an EPSP, an excitatory postsynaptic potential. In the neuromuscular junction, we call them end plate potentials, or they can be a miniature end plate potential. We normally get about 100 of the vesicles releasing. There's about 1,000 or so docked, and about 100 of them release. So you multiply 100 times 0.5, that gives you a depolarization in response to an action potential of about 40 or 50 millivolts, much higher than the threshold to open the voltage-gated sodium channels. Okay, you know that they need to get up to about minus 55 or minus 50. So if we're sitting down at minus 80, this is going to get us well above it from just one impulse in one axon. The way that we finish the end plate potential, okay, because we've released all this neurotransmitter, how does it stop? Why do those ligand-gated channels close? At the neuromuscular junction, the main effect is that we've got an enzyme called acetylcholine esterase, which breaks down the acetylcholine. Okay, so acetylcholine's released, some of it binds, some of it's broken down by the acetylcholine esterase, stuff which is bound releases, and then mainly gets mopped up by the acetylcholine esterase. The breakdown product of the acetylcholine esterase is the choline. Most of that's taken up by the nerve terminal, by co-transport, and then it's used to synthesize new acetylcholine. Okay, so it's kind of wasteful. We have to release it, we break it down, we take it back up, we have to resynthesize it, repackage it, but that's what's happening every moment. Every breath you take, your muscles are doing that. Okay, they're breaking down neurotransmitter, reuptake into the nerve terminal, reforming acetylcholine, reuptake into the terminal, march it down, just to keep us going. Central nervous system termination tends to be a bit different. we get desensitization of receptors and there's glia around to do things. Here at the neuromuscular junction, the main effect is this acetylcholinesterase. Okay, and to remind you about what happens next, this ties in with the action potential lecture and something you'll get from Dr. Head soon. Here's the impulse in the axon. Here's the end plate potential recorded at the neuromuscular junction. So, like I said, there's lots of names. The end plate is another name for the postsynaptic side of the neuromuscular junction. So, here we are at the neuromuscular junction. We get a big depolarization. There's a small delay because when we have the stimulus of the axon, we need to have time for voltage-gated sodium channels to open. We need to have the fusion of the vesicles. We need to have diffusion of the acetylcholine. So that's here the fusion, the diffusion, activating the receptors. There's a small delay. When we look further away along the surface of the muscle, that potential that we record gets smaller and smaller. Okay, here we had neurotransmitter opening those channels. As we go further away, we're getting passive spread of that depolarization across the surface of the muscle. Okay, this is done in a paralyzed muscle, so the voltage-gated sodium channels have been blocked, so we can see what it looks like. If we've got voltage-gated sodium channels, this depolarization would be big enough to start an action potential, and that would start an action potential here, which would propagate to here, which would propagate to here, so we'd have an action potential relayed across the surface of the muscle fibre. Okay, and then that's going to dive in, trigger calcium release, and cause the actin and myosin start interacting. Okay, so although it's simple compared to the central nervous system, there's lots of things that can affect the neuromuscular junction. Some of these you'll know already. So tetrodotoxin, that's pufferfish toxin. Saxitoxin, another toxin I think from a mollusk, they can affect the sodium channel. Dendrotoxin can affect the potassium channel, voltage-gated, which again is part of generating the action potential that comes from a snake. Conotoxin is from cone shells. They're up on the barrier reef. They're pretty nasty. They can block your voltage-gated calcium channel. Of course, if you can't get calcium in, you don't get vesicle release. So all of these things are bad. Negatives mean that they're blocking. Tetanus toxin and Botox we talked about already. So they're all things that can affect the presynaptic side. On the postsynaptic side, there's quite a number of things. The first one that we probably should think about is the acetylcholine receptor. So acetylcholine obviously activates it. Nicotine does. That's why nicotine is banned at the Olympics and so on, because it actually potentiates the acetylcholine receptors. You get a stronger response. You get a bigger contraction of your muscles because you have more depolarization coming through these acetylcholine receptors. Things which block it include tuberculari, which is from the poison arrow frog, famous in World War II spy movies. Bungara toxin from snakes. We can also attack the acetylcholine receptor ourselves. So some people have an autoimmune disease called myasthenia gravis, and what happens there is that you generate antibodies against your own acetylcholine receptors. Okay, and so in different people it works differently. In some people the antibodies block the channels from opening just by binding. It means that the acetylcholine can't bind anymore. In other patients, the antibodies actually cause destruction of the receptors. But one way or the other, you haven't got enough functioning acetylcholine receptors. That causes muscle weakness because you're not getting the depolarization in the muscle. So the first symptoms are usually that they can't keep their eyes open or their speech becomes slurred, often attacks eyes and mouth first, but it can become life-threatening if it spreads to affect the lungs. Then you can't inflate your lungs anymore and you'll die. So those patients need to be put on a respirator then until they recover from that bout. Like lots of autoimmune diseases tends to get worse and get better. So it's not usually fatal. You can live with it, but you need to be very vigilant. And one way to treat it is by attacking the acetylcholinesterase. So acetylcholinesterase breaks down your acetylcholine. That's normally good, but if you don't have many acetylcholine receptors, you actually want your acetylcholine to hang around. So you can take drugs which attack the acetylcholinesterase. Okay? Too much of those drugs are poison. So this is a sort of fertilizer, an organophosphate. This is from plants. The common way is the bug sprays you use on insects mainly attack their acetylcholinesterase. They have too much depolarization and die. But with patients, the right dose can actually keep them alive if they have myasthenia gravis or if they've got poisoning by one of these other drugs. And then finally, of course, the muscle sodium channels get affected by all the same things as our neuronal sodium channels. Okay, so lots of different things that interact here at the neuromuscular junction. Okay, so quick summary of the main three sorts of synapses that we have. Neuromuscular junction in the periphery, central synapses, and then the autonomic, again also in the periphery. Neuromuscular junction is always excitatory. Okay? There's no inhibitory neuromuscular junction. It's always ionotropic. Okay? Because the only receptor involved is that nicotinic acetylcholine receptor. There's a lot of active zones, which means we're going to get a very big response, a big EPSP. We call it EPSP here. Central nervous system, you can have an excitatory or an inhibitory response. You can have ionic receptors or you can have metabotropic, which is second messenger coupled receptors. A typical axon might have about 2 active zones, which means that a typical axon causes a very small EPSP, and you need multiple neurons to come together to produce a response. And that's what you want. You want to be integrating information before you fire the next neuron. Autonomic system, we can be excitatory or inhibitory, and because it's not concerned with speed, uses metabotropic receptors to do that. Okay, so it uses acetylcholine for some of it, but it uses a muscarinic, not a nicotinic receptor. It's got varicosities rather than active zones, and each varicosity potentially can release multiple vesicles, so it can have very big effects. Okay, we don't usually measure an EPSP because these metabotropic effects are much slower, but certainly one autonomic axon can produce a big effect on the muscle that it's affecting. Okay, it can cause contraction of peripheral blood vessels or a bladder wall muscle or whatever. Okay, so we have to talk a little bit about carpal tunnel syndrome because it comes up in the scenario, so that gets tacked on here. Carpal tunnel is a disease that specifically affects the median nerve. There are 2 main nerves that innervate the hand, so the thumb and digits 1, 2, 3 and half of 4 are innervated by the median nerve. The ulnar nerve innervates digit 5 and half of digit 4. The median nerve passes here under the flexor retinaculum, or the transverse carpal ligament. And what that means is if you do a lot of wrist flexion, you can potentially aggravate the median nerve. Okay, it can also happen if you have a fracture which damages the wrist. That's quite a common complication, is that you're going to get impact on the median nerve because of the swelling that's there, or if you've got edema for other causes, that swelling can again compress the median nerve against this ligament. We see, if we look at it in cross-section here, the median nerve is sitting trapped here above the tendons and under this ligament, so it's particularly vulnerable if there's a bit of swelling down there in the wrist. The symptoms of carpal tunnel syndrome are that you get paresthesias, you get pins and needles in the innervation of the median nerve. Tends to be most common when you wake up during the night or in the morning because you've been lying flat, had a chance for fluid to accumulate and for some swelling to occur in the wrist. Okay, some pins and needles doesn't seem too bad, but if you don't treat it, the symptoms can become worse. Ultimately, you'll get axonal loss, you'll get numbness, and you'll get wasting of the thenar muscle, so your thumb will lose its strength as that muscle wastes away. How does that happen? Well, there's a couple of different effects. It tends to be more common in women than men. We said it happens at night. The big axons are more vulnerable than the small axons, so that's bad. You can become numb, lose your sense of touch, but still have pain from it because the pain fibres are small and some of them are unmyelinated. We think that the effects are that the pressure acting there at the carpal tunnel decreases the blood flow inside the nerve and it interferes with axonal transport. Okay, so it's got a twofold effect. One is that it's compromising the blood supply, the other that it's affecting axonal transport. And in the long term we can get degeneration, like I said, leading to muscle wasting. These are the sorts of axons we find inside the median nerve. They come in three sorts: sensory, motor and autonomic. We'll talk about these a bit more in the lab. There are some big myelinated fibres. In sensory they're for touch and position. For motor they're for flexing our muscle. We've got some small myelinateds which handle cold, pricking pain, spindle receptors inside muscle, and preganglionic autonomic axons. And then finally we've got unmyelinated, which are responsible for heat and pain and for postganglionic. Okay, so we've got a mix of different axons inside the median nerve, and these big ones are more likely to be affected because they tend to be more active. The blood supply side is reasonably easy to understand. We'll finish up by talking briefly about axonal transport. So as a neurophysiologist, I spend most of my time thinking of axons as very fast transmission systems. They can send an impulse down here in 50 milliseconds or so because they use electrical signalling. But they do need to send proteins down the synapse, the tagmin, all those V-snares. They need to be sent down to the terminal. They're made up in the soma, the Golgi apparatus, packaged into vesicles, and then they travel down the microtubules carried by particular molecular motors. Okay, so we've got transport going down towards the terminal, which we call anterograde transport, and we've got retrograde transport from the terminal back to the soma. These are pretty, well, depends how you think about it, 400 millimetres a day. That's pretty fast if you're a protein. It's pretty slow if you're trying to go anywhere between Sydney and Parramatta or something. But 400 millimetres a day means that it can get almost from my spinal cord out to my fingers, you know, in a day or two. A couple of days, you'll be able to transport something out there. There's also a slower anterograde transport which moves the big proteins, cytoskeletal proteins and soluble proteins. So the ones that are packaged into vesicles go quite fast using little kinesin motors. The ones that are free travel much slower, 2 millimetres a day. Okay, so to travel a metre down to my fingers takes a couple of months for those things to get there. If we look at retrograde transport, we need to bring back trophic factors from muscle. Again, we've got things packaged in vesicles. They can move quite fast along the microtubules, about 250 millimeters per day. That can be good. You can be picking up trophic factors from muscle to keep your nerve terminals alive, but it's also the way that viruses, such as herpes simplex, which gives you cold sores, it moves up and down your peripheral nervous system. When it's hiding out, it's up in the ganglion. It gets transported down to the endings when you have a cold sore. You can infect other people, travels back up using retrograde transport to hide out in their soma, and then comes back down again with anterograde transport. So mainly for good, but some pathogens have harnessed this system to their own ends. The way these vesicles work, we have two different proteins. We've got one called dynein that binds the vesicles. It walks backwards along microtubules. We've got kinesin, which walks forward along the microtubules. And they appear to have feet which bind and unbind and actually let them do that walking. Okay, and a different protein is responsible for the movement in the different directions. Alright, so what you need to remember from this lecture, you need to explain the sequence of events at the neuromuscular junction, starting from an action potential in the axon, all the way back to producing an action potential in skeletal muscle. Okay, and in another lecture you'll learn how that causes contraction. You'll be able to tell me a bit about carpal tunnel syndrome and a little bit about extroplasmic transport. Thank you. Hiya. That's ligand gated and that's voltage gated, that's right. If I actually want this, I only got the active. So that's what I said, yeah. Exactly, and that's not enough to spread it. The muscle fibers are huge. Yeah. You need an action potential to spread across the whole thing, trigger calcium entry to get the contraction. Yep. That's right. Yeah, so it's kind of nuts. It's an action potential, then it's chemical, then it goes back to an action potential. Yep. Yep, thank you. Yep, sure. Yep. No, so the cold sores have a base hormone that come out. So what they're doing is when these hormones come up here in the soma, and then the anterograde when they affect someone else, then it travels from the skin back up the axon and retrograde transport to get to the new person of the soma and hide out there and later do the two different people, right? So once you're infected, it's going down here, it doesn't need to come back. So you get into a new person, and I want to go from their terminal up to their soma and hide out. Yep. Yeah, okay. So that's the all or nothing is related to voltage gated study. This is being done by the ligand-gated polymer. And so the reason why this can vary is how many of these come over, how much of a retention. A little bit of retention, you have a little effect, a lot of your retention, everything effect. But once you cross the threshold, then you get that potential across the whole muscle system, voltage gated. The voltage gated is all or nothing, ligand gated is definitely greater. Yep. Yep, EPT. No, the EPT and the EPT are not the active potential. They're the ligand gated. So and it may or may not trigger an active potential. But it can be caused by ligand gated. Yeah. Sure. So you have molecular activity today, all or nothing.
But you need to start the action. Yeah, and so the way that starts is you need a depolarization. It comes by these ligand states. And so the more neurotransmitter you release, the more channels you open, the more postsynaptic depolarization. But because we can't release just one molecule of acetylcholine that comes out of packet, we get depolarization in these size steps, which is called quanta. If we release enough quanta, we get a big enough depolarization, we probably need 15 or 20 millivolts, then we'll get an action. Does that make sense?
What?
So, because we've got 1,000 viscers all sitting there ready to go, some of them spontaneously release. How can fluctuate, or maybe the molecule pulls it too close. So, the tiny release of transmitter all of the time. That's just it doesn't, as far as we know, the most useful function, maybe during development or something, but it's mainly because to have the system set up to respond really quickly, there's a certain risk of firing. No, because it's only adding half a millivolt. It's not enough to bring you up to threshold. You need to have lots of these happening together.
Okay.
That's right.
Okay, great. Thank you.
You got a separate question? Are you right? Okay. What are you like?
So like the first diagram we showed
Yep.
it was just like one particular channel which that platform that is also or are they two different channels? Sodium channel and the
No, they're one. This green thing.
yeah.
It's both a channel and it's a receptor.
Yeah.
And that's why we call it ionotropic. It means it's a receptor channel.
Right. Then how come in this one we've got muscle sodium channel and it's going here?
Yeah. So that's kind of related to what I emphasized before. Make it in the program to show you the slide. So this response is produced by these ligand gated because to start the action potential you need a depolarization. For a voltage gated chemical open, there needs to be some depolarization. It's kind of a chicken and egg. How do you get the depolarization? Once it starts, every sodium channel will open and it started. And the way that we solve that is with these channels. These are gated by the neurotransmitter. When they open, sodium comes in, and then we produce these little depolarizations. If they cross threshold, then we get an action potential. So, the one here. Yeah. The one. No, sorry. Oh no, yeah, this is the one. It's got the. And so this is providing the depolarization to start the action potential done by those cells. Right. Yeah?
That table, I wasn't quite sure what you meant by
Okay. So in these cases we can just measure a depolarization and it's easy, it's either going to fire an action potential or not. The targets of the autonomic nervous system, they don't necessarily fire an action potential. So it's not useful to talk about reaching threshold or not. If you're modulating heart rate or if you're causing contraction of sodium muscle or whatever, it doesn't necessarily equate to the idea of reaching threshold and generating action. So one axon can cause a big effect, but we can't measure it in a simple way. But so the main idea is that one axon in the central nervous system pretty much does nothing, seems to work together. But in the peripheral, either the neuromuscular junction makes the muscle twitch, or in the autonomic nervous system, one axon can actually have a pretty big effect on its target.
Right, and it's sort of like physiological.
Yeah, that's right. Yeah, we found physiological. But you can't. So it's not action potential. There's not one language to describe it.
Right.
So it could be the causing release of adrenaline or whatever. Does that make sense?
Yeah. Actually, what's the same one? When it says 1,000 does that mean 1,000 deadly or
1,000 zones. That's right. And I think the theory is that each zone can only release one vehicle.
Oh.
and the mechanism for that's unknown, but we've got 1,000 zones. Each zone has got lots of vegetables ready to go, so we can get hundreds of them. Because your muscles can contract, thousands of times without.
So a single action potential down the axon will obstruct, and each vehicle contributes.